Hello, everybody. This is Debbie Spoon's NSF CaveNet presentation. Tonight we have our speaker, Ben Hutchins, who is going to talk about dark waters, strange creatures, amazing life in groundwater. And I appreciate Ben being able to do this for us. It's, I know people are busy, but this is such a good thing for our cavers, getting information and experience out there. I'm going to talk about Ben just for a second, and then Ben's going to take over, and that way you guys can hear this wonderful presentation that he has for us. So Ben is a caver biologist from Kentucky, and he's lived in Texas for the last 11 years. Now his caving and studying, in the last 11 years, caving and studying underground ecosystems in Texas. Ben received his PhD in aquatic resources from Texas State University after he had attended Western Kentucky University and American University in Washington, D.C. His academic interests focus on patterns of biodiversity and community structure in groundwater ecosystems, particularly in Texas. As a caver, Ben is interested in cave cartography, project caving, and caving nationally and abroad. So, all right, Ben, what I'm going to do is I'm going to turn this over to you. I've already got to see some of his presentation, some awesome pictures. So this is your format, Ben. Go ahead.
All right, thanks, Debbie. I hope everybody can hear me okay. I'm really excited to be giving this presentation with the National Speleological Society. This is one of my favorite topics. This is going to be a whirlwind tour of the cool and unusual animals that live in groundwater. Okay, so let me give you all a little bit of background about what I'm going to be talking about tonight. So I want to provide first just an overview of the different kinds of groundwater habitats. So as cavers, you know, maybe we're familiar with cave streams and you've seen cave lakes, and those are certainly some great groundwater habitats, but I'm hopefully going to introduce you to some new ones that maybe you're not aware of, or maybe you've never thought about them as habitats for animals. Then I'm going to talk about the animals that live in those habitats. So what kinds of animals do we see in groundwater? And what are the patterns of biodiversity that we see? Where do these animals occur? Where don't they occur? And why is that? Then I'm going to talk about the evolution and ecology of these animals. So why do they look the way they do? What do they do down there all day long? What do they eat? And I'm going to end the presentation by talking about how we scientifically describe these species. So that's a process called taxonomy. I'm certainly not an expert in it, but it's something that I've been working on for the last few years and thought maybe cavers would be interested in that process. And then I'll end by talking about how cavers can be involved in cave science in general and groundwater ecology. Okay, so Debbie gave you a little bit of background about me. I've been caving for about 20 years, doing biology for about the same amount of time. A lot of the work that I do in Texas is focused on the Edwards Aquifer here in Central Texas. So this is a global biodiversity hotspot for groundwater animals. And so a lot of the examples that I'm going to be giving are about the Edwards Aquifer, but I'm going to try to keep the talk kind of general, applicable to anywhere on the planet. So I want to start by answering this question: why study these aquifer animals? I was caving with a guy years ago, and he said, "Why would you study these things? Why wouldn't you study something big and cool like whales?" And you know, I guess whales are pretty cool, but it's the same reason that we, you know, do things that potentially have risk involved, like getting on rope or, at least ripping our pants to shreds, going through tight, gnarly passages. It's because we are explorers, right? We're interested in the unknown. We want to know what's out there. And so from a biologist perspective, there's nothing better than groundwater organisms. These are some of the least studied animals on the planet, and new discoveries are being made all the time. So yeah, they're not as big as whales, but they're still pretty dang cool. Okay, and so with that, let's dive into what groundwater habitats are. Okay, I promise that this is the only definition that I have in the whole presentation. You notice that I'm using the term groundwater animals and not using the term aquifer animals very much. And that's because of the definition here. We define an aquifer as a rock formation, so it's not actually the water, but the rock formation, and it's one that stores, transmits, yields economically important amounts of water. Enough water down there that we would want to maybe pump it out and use it, right? And so aquifers certainly are a great habitat for groundwater organisms, but they're not the only habitat. So we'll talk about some really little groundwater systems. If you've ever been going through a cave and a drip of water falls out of a stalactite and lands on your head, that's coming out of one of these tiny little groundwater habitats. Not big enough for us to use in an economic perspective, but still habitat for animals. These animals can also occur outside of rock formations. They can occur in sand and gravel, and aquifers can too, but they can also occur in soils. So for the most part, I'm going to be using the term groundwater animals, but we'll be talking about aquifers quite a bit. And of course, the best kind of aquifers are karst aquifers. And I like this cross-section here because it shows a lot of the habitats that we as cavers may be familiar with. All right, so we have a surface stream and it disappears underground. You can follow that cave stream, whether it's a vertical stream like a waterfall or it's in horizontal passage. If it's air-filled passage, that's called the vadose zone. And so cave streams are a great example of the vadose zone, probably ones that are some of the better studied groundwater habitats and certainly the most familiar to us as cavers. And maybe you've seen some of these organisms crawling around in the cave stream. So if you follow that cave stream down, eventually you get to the water table, right? And so, you run into a sump maybe or a cave lake. And unless you're a cave diver, you can't really go any farther. So as a biologist, it's good to have some cave diving friends that can go down there and grab those animals that are living in what's called the phreatic zone. All right, that's below the water table where all of the voids are saturated. They're filled with water, and that's also great habitat for these animals. If you're not a cave diver, you're kind of limited to sampling that habitat indirectly. We can go over here to the spring, right, where that water is coming out, and we can put nets on the spring or put traps there and hopefully get some of those aquifer organisms that are coming out. I mentioned those drips coming out of stalactites. What that last cross-section didn't show was this habitat called the Epikarst. The Epikarst is sometimes called the skin of karst. So it's an area of dissolution features, fissures, bedding plane, partures, and small void spaces that are above the kind of main conduit formation, above the cave, right? So these are small conduits. We can't really physically go in them unless we just blast our way through it. But it's a zone of storage and transmission of water, both vertical movement of water and lateral movement of water, and it's home to these groundwater animals. And so again, we can sample it indirectly by catching the water that comes out of those drips and filtering it out and seeing what animals come out with it. So maybe you didn't know that maybe that drip of water that fell on your head had little organisms in it. Most of them are pretty small, but apparently in Brazil, there's even a cave-adapted catfish that comes out of those stalactites in some places. So the Epikarst is an important habitat, but one that's not well studied, at least here in the United States, except for some stuff that's been done out in the Virginias. Of course, aquifers also occur outside of karst. And so you can get things like fractured bedrock aquifers that can occur in all different types of rock types. These aquifers can extend to great depth, and they can have groundwater organisms in them as well. And you can have animals occurring in these sand and gravel aquifers, moving through the small little spaces between the sand and the gravel. These aquifers can be confined or unconfined. So this cross-section says a silty layer, but you can imagine that this is an impermeable layer like clay or something, and so the aquifer beneath it isn't directly connected to the surface, right? It's isolated from it, it's confined. Or you might have an aquifer that's not confined and it has direct connection to the surface. So when it rains here, it infiltrates through that sand and gravel and recharges the aquifer. Now, one thing that's weird about this cross-section is that it shows the water table in a really weird way. It shows the surface stream kind of magically floating above unsaturated sand and gravel, right? And that is not how it works. In reality, the groundwater table would come up there kind of underneath and beside the surface stream. And it actually is another important habitat called the hypereic zone. All right, and so this is that saturated zone underneath and adjacent to surface streams. And it's a zone of surface water and groundwater mixing. So we have surface water going down and groundwater coming up and mixing in this zone. And we can sample it for cave animals or for groundwater animals by driving in a metal spike and pumping out water and sucking organisms out with it. And so this picture on the top right shows some great hypereic habitat, that big gravel bar, right? And so, some of the animals that we find here look just like those animals that you find in the cave stream. And in fact, for those cave animals, the hypereic zone can actually serve as an extension of the karst aquifer, right? So if that water in the surface stream is coming out of a karst aquifer, geochemically it looks like cave water. And for these tiny little animals that can move around in those spaces between the gravel, that's basically like a three-dimensionally complex cave, right? So the hypereic zone can actually be a migration corridor for these animals that can move out of the karst aquifer. Last month, David Culver gave a great talk for the NSS on shallow subterranean habitats, and he mentioned this one that has the most absurd name, the hypotelminoreic. We call it a seep for short, and we don't often think of them as a groundwater habitat, but they are. So this is really shallow groundwater, maybe a meter below the Earth's surface, or maybe a little more, maybe a little bit less. But again, you can see in this diagram that it's perched above a clay layer, and so where the surface of the Earth drops down and intersects the water table, you get this hypotelminoreic groundwater exposed at the surface. There may not even be an outflow. It may just look like a damp, kind of wet spot in the woods. But if you start flipping over rocks and looking through the soil and the organic matter, you can find these animals that look like cave animals. We also have groundwater habitats that in marine environments. These are really poorly studied. It's kind of hard to sample for groundwater under the ocean, but it's there. The exception is there's been some work done on beaches and in kind of shallow intertidal environments, but that's about it, with the exception of work that's done in ankyline caves, which are really cool. I'll talk about them a little bit more later, but these are cave systems and aquifers that have a connection to the ocean, right? And so you get tidal pulses moving in and out, and you get mixing of fresh water and saline water. Sometimes that mixing at what's called the halocline is really dramatic, as you can see in this picture, where you have fresh water that is just kind of floating on top of saline water. And you get groundwater animals that are living in both the freshwater parts and the saline parts. And there's one animal we'll talk about later that can actually move in between them. So really cool and complicated groundwater systems. But There are probably other groundwater systems that we could divide these categories up more, but that kind of gives you just a broad overview. I want to spend a little more time talking about the animals that occur in these types of habitats, animals that only live in groundwater. They can't survive in surface water. Those are called Stigobiots or Stigobiots. And that's kind of the topic of the presentation today. We can learn about the kinds of animals that live in groundwater by looking at species lists for different places. I like to compare Texas and France. And so if we look here, one of the things that we notice when we look at groundwater communities, something that is conspicuously absent is that there are very few insects that live in groundwater. And the reason that that's important is if we go to a surface stream, start flipping over rocks, we see that insects are one of the dominant components for surface water systems. So they're dominant in surface streams, but almost entirely absent in groundwater. Here in Texas, we have about 8 species of groundwater obligate beetles. There are just 2 in all of France. The other thing that you don't see a lot of in groundwater are vertebrates, things like fish, right? So we have 3 species of fish here in Texas and 3 species of groundwater obligate salamanders. France doesn't have any native vertebrate species. I'm not actually trying to pick on France. We'll see in a later slide that France in other ways comes out way ahead of Texas. But there's a reason that we don't see a lot of vertebrates. If you think about these vertebrate animals like fish, they're big, they move around a lot. A lot of them are visual predators. Using your eyes doesn't work very good in groundwater if you don't have a headlamp. And, they need a lot of food. And as we'll see later, there's not always a lot of food down there. So you don't see a lot of vertebrates. What you do see are a lot of crustaceans, right? So here in Texas, things like isopods and amphipods, they make up over half of all of our groundwater species. The same is true in France. And we see this around the world. Crustaceans dominate groundwater communities. You also see a lot of soft-bodied animals like snails, right? 81 species of groundwater snails in France. And you see things like flatworms and segmented worms. For what's lacking in groundwater, you also get these really cool organisms that don't occur in surface waters. So the remipedes, or the image on the left, these are probably the most spectacular example of this. So this is an entire class of animals that only occurs in groundwater. Now, what does that mean? To put it in perspective, all mammals, from bats to people to whales, are a single class of animals, mammalia, right? So the remipedes are as different from everything else as mammals are different from everything else, right? So this is a very unique lineage, not closely related to anything else, that doesn't occur in any surface, marine, or freshwater habitats. They're only in groundwater. The Stygiomysids and the Speleogryphaceans are entire orders of crustaceans that similarly only live in groundwater. So groundwater is a really cool environment to study the tree of life and evolutionary relation among animals and how body types evolved over time, because you get these unique and ancient lineages that don't occur anywhere else. And so where do they occur? So groundwater animals occur around the world. I don't think we know of any in Antarctica yet, but they're probably there. But one of the things that we notice when we look at the groundwater sites is that even our most diverse sites don't have very many species, right? So we kind of have this magic arbitrary number of 20. If a site has 20 species, it's a diverse site. Now, if you ask a stream ecologist, somebody that works in surface streams, they would not be impressed. That would be a very species poor, probably highly polluted or impacted stream. But that's pretty good for groundwater sites. And we can see from this top map, those blue circles, there actually aren't very many places on the planet where you have more than 20 groundwater species living in the same place. So we've got the Edwards Aquifer here in Texas. Bermuda and a few sites in Europe and a single site in Western Australia. That's about it, as far as we know. Now, of course, new discoveries are being made all the time. If you look at that lower map, you kind of see more broad patterns. That species richness seems to be highest in temperate regions. So North America and Europe and Australia, right? You don't see biodiversity peaking in the tropics like you do on the surface, right? We think of tropical rainforest as, these biodiversity hotspots in the tropics. And it just doesn't seem like caves or groundwater habitats in general are as diverse in tropical zones, although we are just discovering new things all the time. And it's changing, right? So you see here, Brazil is a relatively diverse area. It's emerging as a diverse area. 20 or 30 years ago, we didn't really know that Brazil had so much biodiversity. And the same with Australia, right? Australia shows up as a hot spot on this map, and that wouldn't be the case if you were looking at this map 20 or 30 years ago. A lot of new discoveries are made in these places. And other places like, a lot of countries in South America and Africa are entirely unstudied. Maya and her colleagues didn't have any data on these countries to populate these maps with. So tons of discoveries yet to be made in groundwater around the world. It's wide open. Okay, so what drives the patterns of diversity that we see? Right? Why are some places diverse and others not? There are a lot of reasons, and we can kind of categorize them in these three categories, which are the history of a place, the energy that's available, the food to support the animals, everybody's got to eat, and habitat heterogeneity. That just means the different types of habitats that are available. And so let's talk about these. A few examples of how history is important. You can see on the left here that species richness in Europe decreases as you head north. And that makes a lot of sense considering that, big parts of northern Europe were covered in ice sheets and permafrost in the Pleistocene. So if you live underground, it's hard to make a living if the ground is frozen, or, if there's a kilometer thick ice sheet over your head, that's a rough life. We can see the importance of history in Australia as well, but a different story here. So with Australia, no glaciers recently, but there's a long history of increasing aridity, right? Australia has been drying out for a long time, especially in Western Australia. But we see really interesting biodiverse groundwater sites in Western Australia in areas like the Pilbara and the Yilgarn. And the idea is that as Australia was drying out and surface streams were going dry, the animals that lived in those streams had basically two choices. They could go extinct, which, that's just a bad day for everybody, or they can go underground, right, where there's still groundwater habitats. And so that drying out drove these species underground where they adapted to these groundwater habitats. And now there's this really weird and interesting groundwater fauna in the Western Australian desert. Here in Texas, where I am, we see the effect of history as well. We know that in the Cretaceous and early part of the Cenozoic, parts of central and eastern Texas were covered by what are called marine embayments, a shallow sea, right? And as that marine environment subsided or retreated, marine animals would get stranded and adapt to freshwater and groundwater. And so we see animals in the Edwards Aquifer that their closest relatives live in the ocean. These are what we call marine relics. And we see them in other places like around the Mediterranean. Some of these animals, these marine relics, have a really cool distribution called a Tethian distribution. So the geologists in the audience might recognize the Tethian Sea. This was an ancient ocean that existed after the breakup of Pangaea. And so when the Tethys was a thing, Tethian animals would colonize groundwater habitats along the coast of the Tethian Ocean. Then the tectonic the continental plates moved around and the Tethian Sea disappeared, but we can still see the fingerprint of the Tethian Ocean by seeing the distribution of these species that lived around the shorelines of the Tethian Sea. So history is important, but regardless of the history, the animals got to eat something, and so energy is also really important. We can see that again with this example in Europe. The black shapes represent an area of really high diversity in Europe. It's been dubbed the Mid-Latitude Ridge of diversity, and it extends into the United States as well. But the idea here is that this ridge, this area is so diverse because, well, as we already talked about, as you move farther north, you run into the effects of the ice ages, right? Where there wasn't a lot of plants growing on the surface, there was low productivity during those Pleistocene ice ages, and these groundwater communities are basically dependent on those plants growing at the surface, right? You have photosynthesis on the surface. That plant material then washes into the groundwater system where it's food for animals. And that wasn't happening in Northern Europe. But if you head farther south into Southern Europe, we know that there were periods where it was really dry. Parts of Southern Europe are still really dry. And so again, you have the idea that historically you had these times of low energy, not a lot of photosynthesis or productivity going on at the surface. And so this mid-latitude ridge kind of represents a sweet spot. It's not too cold, it's not too dry. And so you've had long-term, relatively constant or consistent high levels of photosynthesis going on the surface, and that indirectly then supports communities underground. There are a few groundwater habitats around the world that are not dependent on photosynthesis. The most famous of these is Movila Cave in Romania. That's this picture on the right here. And these are what are called chemosynthetic systems or chemoautotrophic systems. What that means is that you actually have microbes in these systems that are using energy from chemical reactions like the oxidation of sulfide or metabolism of methane. They use the energy generated from those reactions to make their own food, their own organic matter. So just like plants use energy from the sun to make their food, these microbes are using energy from chemical reactions to make their food. And then those microbes are food for a whole diverse suite of groundwater animals. And we see this in other places as well. It occurs in the Edwards Aquifer here in Texas and a few other places, but not everywhere, right? Not everywhere has a bunch of sulfide and methane and things like that. So the energy that's available to drive these communities is really important. So is habitat heterogeneity, a big word that just means the different kinds of habitats that animals can live in. So if you think about physics, in reality, two objects cannot occupy the same space at the same time. Makes sense? Well, in ecology, if two animals occupy the same space at the same time, they compete with one another. They compete for space, they compete for food, and that competition limits how many species can live there. But if you have a lot of different types of habitats, then some animals can live in the big stream, and some can live in the little stream, and some can live in the Epikarst, et cetera, and you can have more species in a place. And so David M. showed this really nicely for groundwater crustaceans in Europe. That's this image on the left here. So you can see the y-axis is species richness, how many species exist in an area. And where it's highest up here, those are areas where you have the highest range of elevations. So not just high elevation, but high elevations and low elevations. And areas where you have the highest habitat diversity, lots of different habitats, right? So the idea here, elevation range and habitat diversity means more different kinds of habitat for animals to live in. Earlier, David Culver and his colleagues showed a similar pattern in the United States. They looked at it a little bit differently, and they showed that just looking at the number of caves in an area was the best predictor of how many species live there. And they hypothesized that it's not the number of caves per se, but that if you have a lot of caves, you probably have a lot of different habitats, right? So caves are, in this instance, what we call a surrogate measure of habitat diversity or habitat heterogeneity. Okay, so switching gears now, one of the reasons that biologists were drawn to groundwater and cave habitats is because the animals that live there look really weird, right? And so we were curious, right, of how did these animals evolve and what do they do? So let's talk about the evolution of groundwater organisms and their ecology. And specifically, I want to talk about adaptations. How do they live in these extreme environments? And we can think about kind of three different classes of adaptations. Morphology, which is just what do they look like? Physiology, what does their metabolism and respiration look like? How do they deal with low oxygen or low energy? And their behavior. How do they move around? What do they do to live in these extreme habitats? So in terms of morphology, there was this idea that subterranean habitats and groundwater habitats were so extreme that you have something called convergent evolution, which means that no matter what the animal's ancestors look like, no matter its family tree, they all end up looking the same because there's only one kind of type of body that works in these extreme habitats. Everything else went extinct. And in some instances, that makes sense, right? Again and again, we see them converging on eyelessness, right? Or converging on no pigment. So again and again, you see these animals that have no eyes or no pigments. And there's probably a lot of different reasons for that, but you know, the classic reason is, well, you don't need eyes down there. So if you have a mutation that means you lose your eyes, it doesn't hurt you. It's neutral, right? There's a lot of debate about whether that's the mechanism or not. But we also know that the story is not quite so simple because you have a lot of different types of habitats. They select for different body types. And that's what we can see here. So on the top left, this is called Stigobiots. It's like the classic groundwater organism in the United States. If you've seen little white animals in cave streams, there's a good chance it was Stigobiots, right? And it had no pigment, no eyes, pretty long antenna, pretty long legs, classic cave stream animal. On the bottom right, this is another amphipod. So all of these are amphipods, right? They're all kind of related to each other. The one on the bottom right is called Texovakelia. And you can see that it has extremely long legs and extremely long antenna. It's not a cave stream animal. This is a deep aquifer animal that swims around in flooded conduits, right? So it's a different type of habitat. So you see that reflected in a different body. On the bottom left, this is called Engulfiella. And it doesn't have some of those classic adaptations that we think of. It has short antenna and short legs. And has this long worm-like body. And the idea here is that this guy is interstitial. That means that it's wriggling through these small little spaces in between gravels, right? And so it doesn't need long antenna or long legs. It would just get hung up as it goes through these tight spaces. On the top right, this is another amphipod called Seborgia. And we don't really know what it does. But again, it's very compact. It has short antenna, relatively short legs. We think that this is probably an adaptation for really tight spaces. It looks kind of like amphipods that you see living in between sand grains on a beach. And so maybe it uses that compact tank-like body to kind of push its way through those loose sediments, but we don't really know. But what we can see here is that even within these groups like amphipods, there's actually quite a bit of diversity in morphology. If we think about physiology, there are a lot of kind of interesting questions that researchers have looked at. This animal on the top right is called Mexistenocellus. It's an isopod. It doesn't even look like a cave animal. It looks bright red, right? And this is what it looks like in real life. But it's not pigmented. That's not pigment in its exoskeleton that you're looking at. We think that's hemoglobin. And hemoglobin is what makes your blood red. It binds to oxygen. And this guy lives in really warm water. Warm water can hold less oxygen than cold water. And we've actually measured oxygen in some of the sites where this animal occurs at less than 2 milligrams per liter. That's pretty low. That's like a third of the oxygen that you would see in parts of the Edwards Aquifer where you get a lot of other animals, or even less than you would see in some cave streams as well. So we think that this is just a physiologic adaptation to living in these low oxygen environments. It's full of hemoglobin to scavenge every little bit of oxygen that's available in the habitat. On the bottom left is the European cave salamander, the cave olm or Proteus, probably one of the best studied cave animals in terms of physiology. I don't know if this is true or not, but I heard a great story from a cave biologist that said they had one of these in captivity, and it swam over and it sat down on a rock, and it sat there. And it continued to sit there, and days went by, and months went by, and 2 years went by, and then it decided to move again, and everything was fine, right? So that's probably an adaptation to low food. We know that these guys can go years, probably over a decade without food. That's a physiologic adaptation to low energy, right? If you live in an environment where there's not a lot to eat and the food that is there is unpredictable, right? You don't know when your next meal is going to come, then over time you can evolve these physiologic adaptations to deal with that. There are also behavioral adaptations as well. So again, if you're thinking about environments that have not a lot of food available, a lot of these animals are what we call generalist feeders. The idea is that they eat anything they can find whenever they can find it, right? And so as cavers, we're lucky that we're the big guys in the cave environment. On the bottom left is the Mexican cave tetra, Astyanax. It's a great animal to study because you have the surface populations that look like regular fish, and then you have cave populations that look like cave fish, right? So you can compare them. And what researchers have shown is that those cave populations behave more aggressively than their surface counterparts. The idea here is that those surface populations have to deal with larger predators that will eat them. So, they're hiding and maybe feeding at a lower trophic level. But in the cave, you don't have those higher level predators. So they're swimming around, acting all aggressive and bold and feeding at a higher trophic level because you have a behavioral shift as you shift to this different environmental condition. So a lot of these adaptations that we're talking about have to deal with food, right? Resource availability. And so there's been quite a bit of work done in groundwater on food webs, figuring out what these animals are eating. And so what Kevin Simon and some of his colleagues found in Oregon Cave in West Virginia is that, well, as you expect, most of these animals are dependent on organic matter from the surface, plant matter that washes in, but that particulate stuff like sticks and leaves and chunks of organic matter, that doesn't actually make it into the system very far. It gets filtered out. And sure, there are exceptions to that, but what they found was that what was more important was actually dissolved organic matter from the surface. So this is organic matter that we can't see, but it's dissolved in the water column, and it's used by microbes in the cave. So these are called epilithic biofilms. They're microbes that are growing on the floor of the stream and in the cave walls underwater. And then those biofilms are then used by the animals that are living in the stream. I talked earlier about Movila Cave and how it's a very different system because you have productivity that's happening there in the cave itself. And when that happens, it sets up kind of interesting conditions, and it sets the stage for something called niche partitioning. Now, what in the world does that mean? Well, this is a predictable and dependable food source. It's there, and it's there over long periods of time, evolutionary periods of time. And we talked about how animals compete with one another. But if you have a stable and predictable and productive system like this, then animals can start to feed on different things or feed in different ways so that they're not competing with one another. They're partitioning food resources. And so these images on the right are scanning electron micrographs of amphipods that live here in the Edwards Aquifer. So they all live in the exact same place, but you can see that their mouth parts look very different. And the idea here is that we can actually get a sense for how these animals feed by looking at their mouth part. And so some of these amphipods are predators of other animals. Some of them are scraping off those biofilms, and some are filtering organic matter out of the water column. So they're feeding in different ways, but that's only possible because you have a system with a good amount of organic matter, and importantly, it's a predictable food source so that these animals don't have to just eat whatever they can find whenever they can find it. And so we're kind of refining our ideas about what food webs look like in groundwater. So the top title here is a very influential paper from the early 2000s. And they basically argued that subterranean ecosystems didn't have primary producers, they didn't have predators, and everything was just eating the same thing. It was a truncated functional diversity. And that is definitely the case in some systems, like some of those cave streams, but we know that it's not always the case, right? And so we're seeing more and more papers that talk about differing trophic niches. That just means the animals are eating different things. Without going into much detail, these graphs on the right here show a measure of food chain length. And again, without needing to explain what these axes represent, what this research showed was that some of these subterranean food webs, some of these groundwater food webs, not only had predators, but they had predators that specialized on other predators, right? So this is like the great white shark of groundwater, feeding on other predators, right? So what the food web looks like, it depends on where you're at. And I think that a great example of how complex these can be and how much more we have to learn, a great example of that are those ankyline caves that I talked about earlier, where you have fresh water in connectivity with the ocean. And so if you ever get a chance, go down to the Yucatan and swim around in some cenotes. It's great, good fun, and you'll notice that there are a lot of plants that are growing in these cenotes, right? And so this is a cross-section of a cenote. And you can see photosynthetic organic matter, plants that are growing in the cenote. That's food for cave animals, right? It can get washed deeper into the system. It doesn't show it on this cross-section, but you can also have organic matter coming in from the ocean. So my friend and biologist, Dr. Jean Kritschka, has a picture of a shark in a cave, right? So remember, all of these animals that are coming from the surface that find their way into caves are just waiting to die and become food for cave animals, right? So that shark is just organic matter. It's food for cave animals, right? So you have freshwater organic matter coming in from the surface, marine organic matter coming in from the ocean. You have organic matter trickling in from the soil. And then where you have the halocline, where that fresh and saline water meet, there you have those microbes that are doing that weird chemosynthetic action, making their own food out of inorganic carbon. And so you have in the single system at least 4 different sources of organic matter. They vary in space and time, and you have freshwater and marine organisms utilizing it. And the remipede that I talked about earlier can actually travel in between the freshwater and the saline parts of these systems, right? So to say that this is a simple food web is probably unrealistic. All right, but to do these studies, we have to describe the species that are there. And so I want to kind of talk a little bit about that process and use the analogy that it's like mapping a cave. And so again, we want to describe species because we are explorers, right? We want to document the unknown. And the species are the subject of study for most of the ecology and evolution stuff that I've been talking about during this presentation. Right, there are plenty of studies that don't necessarily look at species, but we still use species a lot, right? But you can't do that unless you know what they are. And then also, of course, a lot of the environmental protections that we have are for species, right? So if you think about the Endangered Species Act or funding from state fish and wildlife agencies, most of that is focused on species, but those species really need to be described for that to work. And so how do we do that? Well, first, again, here you go, France's moment to shine. I put this slide in here to illustrate that there are a whole lot of species that still need to be described. I hate to use the term, but we all have heard the term flattening the curve. Well, here are two curves that are not flattening, right? So in Texas, we've been describing species at basically a linear rate since the mid-60s, right? So this curve is not leveling off, it's continuing to rise. In France, they've been describing species for 100 years longer. They've got like 4 times as many species as we do here in Texas so far. And their curve is also not flattening out, right? It's exponential, right? So if you project these curves into the future, you can imagine that the number of described species is going to increase and increase and increase. So a lot of work to be done. So the first step is to find an animal, right? First step in mapping a cave is to find a cave. Cindy is doing a great job finding a cave here. I'm trying to find an animal on the right-hand picture using a net to catch animals coming out of a spring. Once you find it, well, you need to determine whether it's new or not, right? You don't want to resurvey a cave that's already been surveyed, and we can't re-describe a species that's already been described. So we maybe as a caver, you check your state speleologic survey or talk to local cavers. On the right-hand side, we've got Benjamin Schwartz in the time of COVID dissecting an animal, comparing it to all of the other animals that it might be to determine whether it's new or not. All right, so once you find a new cave, then you survey it, right? So you take measurements, you draw pictures of the cave in multiple perspectives, you make notes about it, and we do the same thing when we're describing species. We take measurements, we count hairs and count claws and draw all these body parts and make notes. Once you finish surveying the cave, you import your images into a drafting software like Illustrator. We do the same thing with all of our animal sketches, and we make what are called plates. These are pictures of the animal, just like a map is a picture of the cave, kind of. And then you've made your map. What do? You hide it under your bed where nobody will ever see it, right? No, well, that happens, sure, but better to publish your map, right? Maybe it's in your local Grotto newsletter or the NSS News, and even better if you have some words to go along with it in the form of a trip report that tells about how you explored the cave and what it's like. And that's what a species description is. It has pictures of the animal. It tells how we found it, where it lives, and it verbally describes it. And once you do that, then that animal is valid and exists in perpetuity until another biologist comes along and says that we didn't know what we were talking about and redescribes it. That would be kind of like step 6 when somebody comes along and resurveys your cave. All right. So in all of this stuff, there's a lot of opportunity for caver involvement. And so just some thoughts on that, and hopefully this will spur some conversation and discussion within the NSS. But I highly involve interested cavers in finding a local academic partner. Maybe they're at a local zoo or a university or a state agency, but they're going to know the research need. They're going to know what needs to be done. They're going to know where your samples need to go, because you don't want to collect some stuff and then it ends up just sitting on a shelf forever and nobody uses it. And they're also going to know the local laws that are going to keep you out of trouble, right? I mean, we can't necessarily just collect everything that we see everywhere we go. There are some laws in certain areas, depending on where you are, that are against that. Those local academic partners can also teach you how to hunt, right? Like hunting and fishing, it takes some practice, but you can get an eye for what the animal looks like and where it occurs. Then you can start hunting yourself, and then you can start sharing your knowledge, right? As the local cavers, you know where the caves are, you know where the streams are, you know where the good habitat is, and the chances are you probably have better caving skills than those academic partners. And so you can get down into places that they just aren't able to go and get those important collections that otherwise wouldn't be made. And if you do that, for the love of all things that are holy, please make good labels. Pretend that your sample is going to an alien from the future that has no idea who you are or where you came from, right? So this label here is not enough. Is SLO a cave, a spring, an acronym? Who is JC and AE? Spell out your name. Where is Hays County? There are numerous states with Hays counties. Put GPS coordinates on there. That's much better. And write legibly and tell us what you found. What is the sample preserved in? Is it in 95% ethanol? Is it in some tequila that you had? We don't know, but we need to know, right? So make good labels. And if you do this, then maybe you too can be immortalized with a species named in your honor, right? So these are just 7 examples that I could come up with without even trying, of species named after cavers. And these are not cave biologists or cave scientists. They're people like Charlie Savage here, who saw a cool scorpion on the other side of the stream and braved his way across the river and collected that animal, and now it's named in his honor. All right, so there's a long history of that. Aside from that, there are other reasons to do cave biology. Mainly it's fun, and we are explorers and want to learn more about the underground. You can be part of something bigger than yourself. Many of the studies I talked about today were years in the making, right? It's like surveying in Mammoth Cave or Wind Cave, right? So you can be part of something big. And the information that you collect is key to protecting these animals. I know that the NSS is a pretty conservation-minded organization, but some people are apprehensive about, collecting rare species data. They're afraid that it's going to close the caves, because there are relatively very few examples of that happening, but it has happened. But, as cavers, a caver once told me, caves are ours, right? This is our domain. We should have a place at the table. And so by being involved in the science, you can be involved in the decision-making process. Finding animals can keep caves open. I know of examples of caves that have always been closed to recreation, but they're open for scientific purposes. So maybe that's your only chance of getting in a cool cave. And we can actually potentially have a role in keeping caves open or opening them back up. There are examples of animals that were formerly thought to be really rare until a bunch of cavers started going out and looking for them. And now we know that they occur in other places, right? And so we don't have to be as worried about those caves. But for the relatively few species that do need protection, if as cavers we're involved in the process from the ground level up, then at least we can have a place at the table and make sure that the right decisions are made and they're based off of good data. And so, again, I'm not an expert in this idea, but I hope that it spurs some thought and discussion. With that, I'm going to end my presentation. I want to thank all of the people that took photos that I used for this. If they were good, they probably weren't mine. I tried my best to give credit. If I missed somebody, I'm sorry. But thank you all very much for listening, and I hope we have some time for some questions.
All right, well, thank you so much, Ben. We do have one question. Those of you who have questions, please go ahead and use your toolbar or go to webinar and type it in. We had one from Andy Edwards. It says, I assume no eyes is an advantage to this creature, not just a neutral, since producing eyes would be a waste of energy when they can't be used for anything. Is that correct?
Yeah, so Andy, that's a great thought. And I'm going to get over my head in this really quickly if I'm not careful. So the thought on that has changed over time, from it being a neutral mutation to nearly neutral mutation, and there are lots of there's some evidence that it does have a benefit, and that benefit could be that we originally thought that the benefit was energy saving, that you don't waste energy making eyes, then you can use that energy for something else. But it could also be more complicated than that. And I could get over my head with that I think there's some evidence that the same mutations that cause eye loss can actually be involved in other adaptations, such as fat storage or the development of other sensory structures. So there could be a benefit to the animal that's not just cost savings. It could be that single mutations can have multiple effects, and so there's a lot of active research going on that, and it's over my head.
Okay, great answer. All right, anybody else have any questions? I think you've done a good job at explaining a lot of things. Very incredible pictures.
Thank you.
They use microscopes, or how do you get that? How do you get those?
Yeah, so a lot of the pictures that we're seeing here are through a microscope, and that's everything from holding your cell phone up to the eyepiece of the microscope to using fancy microscopes that have built-in cameras. Some of these, I had those scanning electron micrograph images from, those are $100,000 or multi-$100,000 pieces of equipment that take these super fancy pictures. But some of it is just a phone.
Another question from Catherine says, in the Edwards Aquifer, what groups seem to be the most underexplored or undescribed?
Yeah, so in the Edwards Aquifer, the copepods and the mites are probably the least described. So this is a copepod here. And if we go back to like that France versus Texas example, France has, I don't remember how many, 50 or 60 species of copepods or something like that. We have 2 in the Edwards Aquifer. And it's not because they're not there. We get them in nearly every sample, but it's because we never had a copepod specialist looking at copepods in the Edwards Aquifer. So we know that from around the world, copepods are a very common major component of groundwater communities. If there are any copepod biologists listening, please, we have your samples.
Okay, great. Another question came from Sarah. Says, I apologize from the onset of this question. My job has made me a data freak, and here's the question. Can you talk a bit about how your log, okay, I can read. Can you talk about how you log and preserve this data? What does your data set look like?
I guess that depends on
the appropriate way to log things.
Yeah, so that's a great question, actually. I always urge that people that are engaged in research should publish their data, and so publish a paper, but also data to go along with it. But even if it doesn't get published, if cavers are going out and collecting information or collecting specimens, especially, making sure that those go to your academic partner, they'll get them into museums. And more museums are modernizing their collection data. And so making sure that those collection data are available online or at least available digitally. And so that's one of the things I would say that's most relevant for cavers is making sure that it's going to a museum that has like a data management plan. And most museums have that. Most, legitimate museums do.
What would your, the label you showed us that didn't make any sense and you couldn't figure out how that looked. What would yours look like? How would you, if you had your druthers, what would you think it would look like?
Oh, the cross section of the aquifers.
The data you showed us a label that you didn't know what county, you didn't know what state.
Sure. Yeah. So let me zoom in on that. Let me get there real quick. So here we go. So instead of SLO, well, a couple of things. The best way to do this is with what's called archival ink. So you go to your local hobby store in the art section, and they have these archival ink pens. A pencil is fine for years, but eventually it can fade. So that would be my number one thing. Use an archival ink pen. The name of the cave. So this would say Slow Cave, or if SLO is something else, Slow Spring or Slow whatever. And then I would say the animals that you found, if you know what they are. So maybe you don't know. Maybe it's aquatic cave bugs. Right, as much as you know, write it down on there. How many? So 6 aquatic cave bugs. Collected by, so spell it out. Collected by your name and whoever else was on the trip, and the date. Don't use numbers. Use the month like they did here, because in Europe and North America we do things differently. So you might collect something here in North America and they send it to Europe, and you wrote 3/4/17, and they think that is the 4th of March or the 3rd of April, right? So spell out the month, and I would say Hayes County, Texas, USA. So spell out everything, and please give GPS coordinates. And in the chance that these people are listening, this is actually just an example. I know exactly where this came from because we work with these people all the time. So this was actually just fine. But it was a good example. If I didn't know where this was from, then it'd be problematic. And you can write on the front and back. Also put the label inside of the sample. So a lot of times people want to tape it to the outside. The problem there is that labels can fall off.
Mm-hmm. Okay, and then where does that information go?
So hand it over to your academic partner. And so here in Texas, for example, for decades, cavers carried around little bottles of alcohol in their cave packs, and whenever they stopped, they'd collect animals. And they'd give that all to James Riddell. He was like the grandfather of Texas biology, and he would painstakingly curate all that stuff at the University of Texas Memorial Museum. And then now all of those data are being entered into the UT Biodiversity Collections database. So you as a caver don't necessarily have to take on the burden of keeping all this information yourself if you have a plan for where that stuff is going to go after you collect it. Okay, and if they don't know anybody, how do they find somebody? They just find a university and do some asking, or how do they basically set it up? I would. This is like my NSS grotto cheerleading answer. I would start in your local grotto. Ask around in your local grotto if other cavers know of cave researchers. If that leads nowhere, the NSS has a cave biology section, so you can reach out to folks in that section. If not that, start doing some Google searches for articles in things like the Journal of Cave and Karst Studies, or there are other academic journals like Subterranean Biology, you know, and doing some of those Google searches can get you places if the NSS, if your grotto or the biology section comes up short.
Awesome. All right. Any other questions for tonight? If there aren't any other questions, but you think of questions later, please go ahead and email me, and I will forward it to Ben, and he can answer them. On your last slide, did you have your email address, Ben?
I did not actually, or did I? Maybe I did. Let's see. Good question.
Because
no, I did not. But I am happy to share it. Although I didn't include it on my slide, but I am happy to share it.
Okay, you want to put your other slide back up? There you go. Yeah, if you could tell people spell out your email address or something so that they can get it. And
how about I type it into the chat? Is that?
And then put, dispense to everybody. Yep.
Okay. Yeah. I got you.
That would be great. And then if you could spell it, because the chat is not does not show up on the recording. If you could spell it, that would be great.
Here, I'm having issues with that, so I'm just going to write it right here. Can you see that?
Yep.
There we go.
Sure can. Okay. So it's Benjamin Hutchins, B as in Ben, H as in Hutchins, 1333 at txstate.edu.
Yep. There it is.
All right. Well, Ben, we've kept you long enough. Thank you very much. This was very amazing, informative, and also great pictures. All right, everybody.
Thanks, Ed.
Thank you for your time. This has been a great, awesome webinar. Questions, comments? We already have thank yous coming in saying thank you. They learned a lot tonight. You've kept the audience's attention very well. My controls tell me that.
Oh, thank you. I had a great time giving the presentation.
Hey, you did a great job. All right, we want to thank the NSF, our parent organization, for sponsoring this. Without them, a lot of cavers be more in the dark than what we usually are. Everybody, please be careful. Be aware and take care of yourself. We're getting closer and closer to the end of this pandemic. Don't give up now. Keep your guard up. Be safe. And we'll see you next time. We have a webinar. Everything's moving to Zoom. All webinars are recorded and stored on the NSS website of caves.org. That's caves.org. If you look on the left side of the screen, you'll see webinars. We have well over 50 webinars now. You can download, you can watch online, you can share them with anybody you choose. It's great for education purposes. All right, everybody, thank you very much. Cave safely, and we'll see you soon. Have a good night.